Thank you very much for your invitation and welcome everyone. I am glad to talk about this topic, the SARS-CoV-2 variants and how to capture them. And to get started, I work at Cerba laboratory, which is a specialized medical laboratory with a platform and a unit of next generation sequencing for genetic and infectious disease. As soon as May 2020, we work on a SARS-CoV-2 sequencing solution, or more exactly, on a respiratory viruses sequencing assay. Our objective wasn't the clinical diagnostics because it was useless, as we had efficient tools like multiplex RT-PCR or qPCR with a capacity of 10,000 tests daily. We wanted to be able to offer a solution to pharmaceutical companies and public institutions monitoring and studies or clinical trials of new antiviral treatments and vaccine candidates. In practice, over the course of this pandemic, SARS-CoV-2 sequencing is useful to investigate viral crisis spaces, the disease epidemiology, to investigate clusters or reinfection cases, and to check RT-PCR assay performances, as we will see later in this presentation. First, let's have a look on the COVID-19 worldwide epidemic. As we can see on this slide, 3 successive waves occurred, and each of them could be linked to a new variant. In this figure, which represents the frequencies of circulating lineage of SARS-CoV-2 over time, the first wave is linked to the clade 20A and 20B, the second one in Europe to the clade 20A EU1, and the third one to the B.1.1.7 lineage, also called V1 or UK variant. But understanding which variants are concerning and why requires an appreciation of virus evolution and the genomic epidemiology of SARS-CoV-2. First, what is a variant? A variant is a SARS-CoV-2 genome which differs from the reference virus, the Wuhan virus, in its nucleic acid sequence. These differences, called mutation, arise as a natural phenomenon during the replication. And if mutation that confer a competitive advantage with respect to viral replication, transmission, and escape in immunity, this mutation will increase in frequency. In 2021, the World Health Organization gave a classification of variants according to criteria about their impact on transmissibility, pathogenicity, and immunity response. The VOC, variant of concern, are variants with evidence of an increase in transmissibility, more severe disease, significant reduction in neutralization by antibodies. The variant of interest are variants with specific genetic markers that have been associated with changes to receptor binding, reduced neutralization by antibodies, and/or reduced efficacy of treatments. The third one is variants under surveillance, which are variants with specific mutations found in VOC and VOI, but without evidence of disease impact or transmissibility. So far, impacting mutations are almost exclusively in the spike protein, on the receptor binding domain and the N-terminal domain, The receptor binding domain interacting with the cell receptor, and the N-terminal domain with the host immunity. As you can see on this slide, in addition to the mutation in spike, some mutations occur in other parts of the genome. That's why it is very important to have the whole genome sequence, in order to classify variants into clades or strains. Hence, get the 29,903 nucleotides of SARS-CoV-2 genomes requires efficient sequencing strategies, enabling the retrieval of the maximum number of high-quality and full-length genome. By next-generation sequencing, two target enrichment protocols are possible: the capture-based approach and the Amplicon-based approach. The Amplicon-based approach is faster and easy to handle, but it is subject to genome variation and lack of hybridization of primers if there are mutations at primers position. It can lead to sequencing gaps and hence inconsistent in the elucidation of new SARS-CoV-2 mutation. The capture-based approach is longer and more expensive, but it is not subject to genome variation, as it tolerates up to 10 to 20% of mismatches. We at Cerba Lab, we chose the solution by target enrichment according to Twist Bioscience and the assay, the Twist Respiratory Virus Research Panel. We chose a method by target enrichment by capture probes, and it is possible to search for 29 common human respiratory viruses thanks to its 41,047 unique probes. We use the Illumina sequencing technology, and our IT solution for the assembly methods is nanoware software by Life&Soft. After a few optimization points, we will talk about on the Q&A sessions if you want to. We obtained expected results with an excellent specificity, as you can see on this table, between the SARS-CoV-2 and a virus very close to the SARS coronavirus virus. A good sensitivity and a genome coverage and a coverage depth good enough to respond to the ECDC guidelines in terms of transmission patterns, clade lineage assignment, detection of unknown pathogens or highly different strains, or to search for quasi-species haplotypes. Here are presented our bulk results from direct next-generation sequencing of human nasopharyngeal swab from COVID-19 positive patients we had in our lab. Cycle thresholds inferior to 32 by Perkin-Elmer RT-PCR and indexed samples grouped by virus concentration in each pool capture are essential. As you can see, 90% reads, we obtain 19% reads with a quality above 30 by Illumina MySeq, and 90% read with a quality above 28 by Illumina NextSeq. Genome sequencing and variant calling were obtained for 95% of samples with a full coverage above 20 and 81% samples with a full coverage above 100. But which sample do we sequence? As it is recommended in the Guide to Implementation for Maximum Impact on Public Health by the World Health Organization, we follow our national indication. We sequence to identify the geographical distribution and the rate of different variants by random sampling. We sequence to identify new variants, cluster sample sequencing, infection despite total sham vaccination, and reinfection cases sequencing or long viral shedding in immunocompromised patients. In November 2020, as you know, only 0.09% of French samples were sequenced and shared in the GIZ platform. Since January 2021, the French Public Health Authority implemented flash surveys every 15 days on Tuesday in order to sequence 15 to 25% of positive RT-PCR. By twist, and depending on flash survey, Cerba performed up to 55% of sequences. It is thanks to its network of more than 450 medical laboratories in France. Most are nasopharyngeal swabs from outpatients. That's why it is therefore representative of circulating variants all around the country. In January, the first SARS-CoV-2 sequence from lineage B.1.525 included in GIZ was characterized by Cerba Lab. In the late January, the only two sequences identified in flash survey as V3 strain, also called Brazilian variant, were found from our network samples, and in April, 58% of the SARS-CoV-2 wide variability was present in Paris area. The second indication is to investigate cluster in order to develop specific countermeasures in health facilities when several cases occurred closely together. It is very useful for this indication, but also in February 2021, you can see that different we show that it were different clades in a nursing home and geriatric unit, which was the causative infection from residents. In Mayenne Hospital also, 3 distinct contaminations as 3 variants were found. In March 2020, 17 elderly people were infected by the V2, also called the South African SARS-CoV-2 strain, and 13 out of the 17 people was received 2 vaccine injections, a total sham vaccine. The only one who was died is one unvaccinated patient. The other indication for us, but not for the Public Health Authority, perhaps also, it is to improve diagnostics. There are a lot, as you can see, there are a lot of mutations all along the SARS-CoV-2 genome, and if they are present on primers and probes design, they affect PCR performances. To counteract these pitfalls, at least 2 targets among gene target gene, ORF1, A, B, S, or E, and N genes, are needed in each PCR assays. We found atypical results in our RT-PCR assays performed in our network, and we can distinguish several profiles. For the atypical profile in RDRP, 3 patterns of mutation is in the N gene within different clades, and other one which is in progress yet. We also use NGS in order to compare commercial screening assays with SARS-CoV-2 sequencing. Screening assays are second intention PCR in order to look for specific mutation as the 501 or the 484. And as you can see on the left, we compare with from the to the gold standard, which is the sequences of our swab. There are very good results for these 3 assay with a statistical kappa between 0.8 and 1. On the right, you can see atypical profiles with a low signal amplitude of the deletion 6970. That's why we wanted to confirm by sequencing that this swab are with the B.1.1.7 variants in it, and the sequence are really this UK variants. To conclude, Twist Respiratory Virus Research Panel is one of the next generation sequencing commercial solution to detect and characterize SARS-CoV-2 variant with strong results on nasopharyngeal swab. We want to try to sequence with another specimen, the saliva, and it is yet in process. In 2020-2021, no other viral pathogen was responsible for respiratory winter outbreaks, as you can see in this figure. But we decided to keep on with the Twist Respiratory Virus Research Panel in order to be ready to offer a solution for monitoring studies for all these common viruses, respiratory viruses, in order to try to investigate new antiviral treatments and vaccine candidates. Increased importance of virus genomic sequencing for clinical and epidemiological investigations. And up to, us to design molecular assays in order to prepare strategy to counteract future emerging disease. Thank you very much for your attention, and thank you to our Cerba team and Life and Soft team.